At Lung Bioengineering, we're deeply concerned about the lack of suitable lungs for transplant. Ex Vivo Lung Perfusion, or EVLP, is a technology currently undergoing a clinical test to determine whether it can prolong the health of lungs that might normally be rejected. Our LB-1 facility in Silver Spring, Maryland, is designed to be the first full-service EVLP center in the United States. The building houses everything necessary to perform EVLP. As we begin our U.S. clinical trial to study this technology, we'd like to invite you to learn more about how our EVLP trial process works. The process begins when an Organ Procurement Organization, an OPO, generates a match run list for suitable organs from a deceased donor. The OPO utilizes DonorNet to electronically notify transplant centers and make offers to the different transplant centers' potential recipients. Allocation continues until a recipient is located. In about 25% of the cases, lungs are placed for transplant. If a transplant surgeon at one of our participating trial centers accepts a lung but considers it marginal, they'll direct it to the LB-1 facility for EVLP. They'll communicate the details of the case, including demographics, surgery time, reason for EVLP, and organ quality. If the donor and lungs meet the inclusion criteria for the clinical trial, our specialists work with the supplying OPO to coordinate transportation to LB-1, including air and ground medical transport. Next, the case is initiated in our Case Management Exchange System, CMX. Referral information includes UNOS ID number, date and time of referral, participating OPO, transplant center, and the physician at Toronto General Hospital who will be participating in the case. The blood type, chart, and authorization disclosure form are received for review by the EVLP specialist at LB-1, at which time the procedure room is assigned. When we have an estimated time of arrival at LB-1, our CMX system alerts security of the incoming ground medical transport. The primary EVLP specialist also alerts the LB-1 staff. The entire team of EVLP specialists, QA lead, and EVLP technicians proceed to LB-1. IT performs system checks. The medical director and general manager are alerted for remote monitoring of the case. At the LB-1 procedure room, setup commences, and all case consumables are put in place by the clinical staff following the CMX checklist. During the same time, an IT specialist establishes a video conferencing link for collaborative online meeting during the case. Next, the OPO coordinator or recovery surgeon communicates donor cross-clamp time to the primary EVLP specialist. Shortly thereafter, the lung is explanted and packaged according to UNOS protocol and loaded onto ground medical transport or transportation to the airport. Once the transport arrives at the local airport, the lungs are transferred to the aircraft. There is a continuing communication between the primary EVLP specialist and the medical transport provider, including flight status and updates on the ground medical transport status. When the flight arrives at the airport local to LB-1, the lungs are transferred to ground medical transport. As soon as ground medical transport is en route to LB-1, the sterile field is established, and the EVLP specialist scrubs in. No time is wasted. The lungs are received at LB-1 and checked in. This includes checking the UNOS number and confirming that the correct lung is going to the assigned procedure room. At this point, the CMX system automatically sends out a notification to the participating centers, including the OPO, the transplant center, LB-1 clinical staff, and the medical advisory team at Toronto General Hospital. The lungs are moved to the procedure room and packaging is inspected. Pictures are taken of the internal and external labels. Then blind verification of the UNOS number and blood type are completed in CMX. Then the lungs are removed from the shipping container and moved to the sterile field. A back table evaluation commences, noting organ condition. Cannulation, intubation, and back table flush are performed. A pre-EVLP biopsy is taken. The perfusion circuit is then primed, and the heater cooler is connected to the heat exchanger. Then lungs are transferred to the procedure table by EVLP specialists. Pressure lines are zeroed, cannulas are connected to the perfusion circuit, and air is purged from the line. The perfusion pump is started, and with this step, the EVLP procedure clock starts. Perfusion continues over a 3 to 6 hour period with ongoing CMX documentation and assessment steps. These include bronchoscopy, X-rays, and blood gas analysis, both on a regular and an as-needed basis. Throughout this period, the IT specialists in our command center facilitate visual and data communication with Toronto General Hospital and the transplant surgeons, keeping them informed on an at least hourly basis. At the 3-hour mark of perfusion, the collective team makes an initial assessment of the lungs. If the decision is made to go ahead with transplantation, the primary EVLP specialist contacts the transplant center to arrange transportation. After all the data is collected and the Toronto General Hospital surgeon and transplant surgeon agree on cessation of EVLP, the case is locked and a disposition email is automatically sent to the Toronto General Hospital surgeon, the transplant surgeon, and finally to the OPO, which confirms transfer of custody to the transplant center. Cooldown commences, and once the cold temperature set point is reached, cannulas are clamped and the lungs are disconnected and moved to the back table. The EVLP procedure clock is stopped. The lungs are then flushed with steam and stored in cold preservation solution. The EVLP specialists remove the cannulas, staple the trachea, and perform a post-EVLP biopsy. LB-1 clinical staff then prints out the CMX perfusion record for inclusion in the shipping container. The organ is then packaged according to OPTN policy. Pictures are taken of internal and external labels. Original tissue typing materials are included in the packaging with the original donor chart and perfusion record. The organ is then brought to the loading dock by LB-1 clinical staff for transport. Once loaded onto ground medical transport, the trip to the airport begins, and the transplant center is advised of departure from LB-1. The post-procedure checklist is initiated in CMX to close out the case. Ground medical transport arrives at the airport and the lungs are loaded onto the flight to the transplant center. The lungs arrive at the destination airport and are transferred to the ground medical transport. Finally, the lungs reach the transplant hospital and are transferred to the custody of the hospital staff by the ground medical transport staff. That concludes our full process, the way the LB-1 clinical trial will operate. Currently, we are working to enroll 8 transplant centers in the U.S. In the end, we will have results for 66 patients whose transplanted lungs have gone through EVLP at LB-1 in Silver Spring, Maryland. We at Lung Bioengineering feel fortunate to have been given the chance to try and reduce the devastating effects of end-stage lung disease and hope our clinical trial will enable us to increase the availability of transplantable lungs. We are always looking towards the future.